Okay, today we will start with DNA replication, and I'm sharing my slides. Yesterday there was some error in this sharing process. You are presenting your screen. Okay, infinity mirror is here, and so it should not be a problem. Okay, so the first thing which we will be talking today is about DNA replication. So when does DNA replication take place? DNA replication, it takes place when the cell divides. When the cell divides. DNA replication takes place. When does the cell divide? Cell divides in two times. Once when it during mitosis, cell division takes place. At that time, in the S phase. In the S phase, there is DNA replication. It also divides in bacteria through this in the process of amitosis. Whenever one cell becomes two cell, one strand of DNA has to become two strands of DNA. So one n amount of DNA will have to produce 2n. One strand will have to become two strands. So this is actually when DNA replication starts to take place. So this is a particular free nucleoside triphosphate. This is the adenine base. The sugar the sugar has been drawn as a straight line. Sugar has been as straight line. This straight line is the sugar. This is the 3' OH. And this is the 5' phosphate. The first phosphate is known as the alpha phosphate. Second phosphate, this is known as the beta phosphate. And this is the gamma phosphate. So this is the nucleoside triphosphate. Specifically, this is DATP. So this is a nucleoside triphosphate. So deoxy it is deoxy adenosine triphosphate, and this is a growing daughter strand. So you have one strand of DNA 5' to 3' direction, and okay, forget about the direction. On top of this another strand of DNA. This acts as a template for another strand of DNA. This is the growing daughter strand. So here you have the 5' phosphate, and to this 5' phosphate you have the sugar backbone. You have adenosine A. Again it is bound to, this is the 3' OH, to this 3' OH. Here was a 3' OH, it is bound. Another triphosphate, deoxy adenosine, or this deoxythymidine triphosphate, deoxyguanosine triphosphate, cytosine triphosphate. And you have this OH. So this 3' OH, it attacks the alpha carbon. So what happens? Eventually, once when this attack takes place, you can expect another this chain to grow. Then another attack takes place. It grows. So gradually the DNA chain keeps on increasing. Clear, everybody? Yes, no?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. So this is the growing DNA chain. The growing DNA chain. Can you see the red dot moving?
Yes,
Yes,
sir.
sir.
Okay. So this is the growing chain, right? Like I said, the sugar is drawn in a as a straight line. This is the, this strand is the template strand. This strand, right? And A C A T C. So the complementary is T G T. So the chain growth, it occurs in the 5' to 3' direction. The chain growth occurs in the 5' to 3' direction. And you can see a triphosphate. This is the alpha. Next one is the beta. Next one is the gamma. And DNA polymerase is the enzyme for DNA replication. And gradually what happens? The 3' OH of another one attacks, and you have this OH formed over here, and this pyrophosphate is removed. This is cut over, cut. Right, 5' phosphate. This is the 5' phosphate, 3' OH, and pyrophosphate. Okay. So this is the shorthand notation. Now there are some models of DNA replication. What are the three models of replication? Semi-conservative mode of replication, conservative mode of replication, and dispersive mode of replication. What is semi-conservative? These parental strands, they separate. These two strands, they separate. You see this pink colored line? Are you able to see this pink colored line?
Yes, sir.
Okay,
Yes, sir.
this pink strand, one strand comes over here, another strand comes, and this blue is the new first replication cycle. This new blue strand, complementary to this first strand, is formed. In the second cycle, what happens? This also separates. This blue gives another blue, and this pink on this, a new strand is formed. This is the second replication cycle. This is known as the semi-conservative. In the conservative model, the whole strand, this one remains same as this, and on top of this, a new double strand is formed. This gives these two strands, and this gives this two strands. The original the duplex, it remains as the same. On top of the duplex, the new strand. This blue colored strand are the new ones which is formed, and this pink is the old one which is formed. And the third model is the dispersive mode, where bits and pieces, bits of this pink old is there, then a new blue is formed, then bits of this or pink is there, blue is formed. So it's like scattered, mixed. So you're able to understand this,
Yes, sir.
the three modes. So Messelson and Stahl, through their experiment, this is for you to do a self study. I will give you you have the materials, how they did the experiment, and you have to ask me the questions. So they have found out that the DNA replication is semi-conservative. They have proved. Now let us see an overview of DNA replication. The first step is initiation. Always there is an origin of replication. There is always an there will be always an origin of replication. Normally in prokaryotes, the DNA is essentially circular, which is present. So when replication takes place, if this is the original origin of replication, replication takes place in two directions. It is a bidirectional replication. If this is like this, one direction the replication takes place here, then if this is the origin of replication, the sequence is the origin of replication, the replication takes place bidirectionally. So therefore, if this is a double strand, the four strands will form. So this double helix is first unwound. So the first step is DNA helicase binds to the lagging strand. The DNA helicase is binding to the lagging strand. So it binds to the lagging strand. Now, what is the leading strand and lagging strand? This I will explain later. And what is the replication fork? So this region which you can see over here, this fork-like region, let me explain you once a few things on a whiteboard first. Able to see the whiteboard?
Yes,
Yes,
sir.
sir.
Yeah,
Yes,
okay.
sir.
Okay. So few things which I have to talk. First is we have to talk about what is leading strand, lagging strand, and what is replication fork. Another thing is replication origin. So if this is a DNA strand, this is a, say, in bacteria normally DNA the bacterial chromosomes, bacterial, you can call them chromosomes. or you can bacterial DNA, they are normally circular. Let us say this region on both the strands, this has a sequence, a specific sequence on which the enzyme for replication DNA polymerase binds. And that is known as the replication origin. Now, let's take a very light color. Let's take this. Let us take this thing over here. You have two strands. Okay, let this be the 3' end, 5' end, and this is the 3' end, 5' end. Are you clear about this region? I'm just taking this region outside separately from this circular part. So the first step will be DNA helicase will bind, say, the lagging strand. Now, what is, let us think this DNA helicase. For the time, forget what is the lagging strand and leading strand. Just concentrate that DNA helicase will bind, and as the name says, helicase, it will unwind the DNA. So this helicase, say the helicase enzyme, one may bind over here, one may bind over here, and the helix will be unwound. Clear so far? Yes, no? Hello?
Yes, yes, sir.
Yes, sir.
Okay. Now, these strands must be kept away from each other. These strands must be kept. If there's something is, if these strands are not kept that is the black strand, this strand and this strand, they must be kept away from each other. If they are not kept away from each other, they will again, they will rewind. These strands are actually, why are these strands needed? They are needed for synthesizing new DNA strands. This particular space need to be kept away from each other. Because in this space, new DNA is being synthesized. If this space is not there, new DNA, there will be no space for synthesis. And for that reason, proteins called SSB proteins bind. This means single strand binding proteins. These are the single strand binding proteins. You will have these single strand SSBs. They will bind and keep the two strands away from each other. Are you people able to listen to what I'm saying?
Yes, sir.
Yes, sir.
Yeah,
Yes,
right
sir.
now, has the whiteboard gone?
Yes. Now, sir, it's gone.
It's gone now, right?
Yes, sir.
Okay. Now I bring back the whiteboard again. You are able to see the whiteboard?
Yes, sir. We will see the
Yes,
whiteboard.
sir.
Okay. Now again, this is right. I'm drawing the same thing. Now when these the two strands are, one is opening in this direction, another is opening in this direction. Right. Here synthesis is taking place from one here, and the synthesis is taking place in this direction. DNA is being synthesized, and the strands are being unwound in this direction. What happens is, stress is built up over here. মাঝখান থেকে খুলছে, so কোনাতে you have stress is built up over in these two regions. the strands can break. So to relieve the stress, there is this enzyme known as DNA gyrase or topoisomerase 2 binds this yellow region. What it does is there are two strands. It cuts one strand and relieves the stress. Relieves stress and joins it again. So this topo 2 actually it removes the stress. Now I'm going back to the PowerPoint presentation. Have we come to back to the PowerPoint presentation?
Yes, sir.
Yes, sir.
Okay, you people can see the PowerPoint presentation. This is what I have talked about. So first step we had talked about the DNA helicase. It unwinds the DNA at the origin. The second I had talked about the SSB proteins. And third I had talked about DNA gyrase or topoisomerase 2. It relieves the stress over here. So this is the DNA helicase, DNA gyrase, and these little proteins are the single strand binding proteins. And unwinding keeps on taking place in both directions. Here replication is taking place. So we know about the initiation of replication right now. So is this part clear? Yes, no?
Yes, sir.
Okay.
Yes, sir.
Yes, sir.
Okay. Now DNA replication has got a thing known as semi discontinuous replication. One thing is for sure, DNA polymerase can replicate only in 5 prime to 3 prime direction. Otherwise, it cannot do so. So you see this blue strand over here, that is the 3 prime end, and this the 5 prime end of the blue strand. And in this, you see the red strand over here. This is the 5 prime end, and this is the 3 prime end over here. So since replication can take place only in the 5 prime to 3 prime direction, there is this strand known as leading strand. There is no problem with it. 3 prime to 5 prime, opposite 5 prime to 3 prime. Since the strands are anti-parallel. Since the strands are anti-parallel. Am I clear? So far?
Yes, sir.
Okay. Now this is actually perfect. It is a leading strand, and that has got no problems. And therefore, this strand, leading strand, in this leading strand, what you find is a continuous replication taking place. But now a problem takes place. In the red strand, this is a 5 prime to 3 prime direction, and you know it cannot replicate in DNA polymerase, cannot do it in the 3 prime 5 prime direction. So it can only do it in the 5 prime to 3 prime direction. So what it happens over here is, when it is opened, the lagging strand, it is discontinuous. First, this part has opened. This strand is synthesized in the 5 prime to 3 prime direction. Then this part opens. First, this part is unwound. Previously, it was all closed like this. Right? This part first unwinds. And in that part, while this leading strand is done, slowly in the opposite direction, this lagging strand is synthesized. Then again, this part is first unwound. Then second, this part is unwound. Then again, from the back, again it comes back to the front. পেছন থেকে সামনে চলে এলো আবার. And again, this little part is formed. Again, this third region is unwound. And this comes over first. So what you have is little discontinuous strands. It has gone back, gone some distance forward, then it has stopped. Again it goes back again. পেছন থেকে এসে আবার it goes ahead. Again it goes back, and then again it comes over here. So first this is made, then again the polymerase goes and makes this, then again the polymerase goes back, and then it makes this. So as it unwinds, so the direction is in this direction, right? So this is known as discontinuous replication, and these little fragments are known as Okazaki fragments. And this part which you see over here, this is known as replication fork. এইটা হচ্ছে replication fork. So this part is the replication fork. Clear? Yes, no?
Yes, sir.
Yes, sir.
Okay. So
Yes, sir.
everybody yes, sir, to? Okay. So here are the components which we are talking about replication for. First, you have talked about the origin of replication. So this is clear. What you now understand what is leading strand. Lagging strand. Origin of replication. Then replication fork. We are clear with it. So this is the origin of replication. Then the DNA helicase. What it does is, this enzyme unwinds the double helix by breaking the hydrogen bond. So they are held together by hydrogen bond. So DNA helicase, it breaks it open. So this is the 5 prime strand, 5 prime to 3 prime strand. This is a 3 prime to 5 prime strand which I had drawn. This is the replication fork. Direct, no problem. This unwinding is taking, the helicase is working like this over here. It is unwinding. And 5 prime to 3 prime direction. This is the leading strand. And then first this is synthesized from this, then this is synthesized, then this is synthesized. We have the lagging strand. And these are the replication fork. Now, one thing to memorize, there is always, before replication starts, when replication starts, this is the 5 prime new strand, 5 prime to 3 prime direction new strand. There is always a little RNA primer needs to be added. A RNA primer needs to be added. Otherwise, replication will not start. And there is this enzyme known as primase. Okay, then we are talking about the enzyme known as DNA ligase. So you have these little Okazaki fragments formed over here like this. There are no gaps actually, but these are not joined by phosphodiester bond. This region, this broken region, has got an OH over here and a 5 prime phosphate. This is the 3 prime OH. This is the 5 prime phosphate. There is this nick over here. This DNA ligase uses ATP, and these little joints, they are attached. The phosphate, these bonds are, this is attached, joint. Nicks are joint. And this is the function of this DNA ligase. Clear? Yes, no?
Yes, sir.
Yes, sir.
Okay. So replication initiation on E. coli. There is a 13 base pair repeat and 9 base pair repeat. DNA A comes and binds over here. DNA helicase binds, it unwinds this region. All the helicases are activated. RNA primers are added. So we have studied over here. So you have the DNA A initiator protein. DNA replication origin is known as ORI-C in E. coli, and to which DNA helicase, that is known as DNA B, and helicase loader, it is known as DNA C. So you have DNA, the name is different. DNA B, DNA C. This is the हेलिकेज. हेलिकस को जो चढ़ाने वाला प्रोटीन, लोड करने वाला प्रोटीन, So let us see what are the events. First is the initiation which is taking place. RNA primer made by DNA primase. There is this enzyme known as primase. DNA primase. It makes the RNA primer. So understand that actually originally Roger Kronberg discovered the DNA polymerase, and that was the DNA polymerase 1. But that is a different function. The real polymerase is the DNA polymerase 3. So first DNA helicase is unwinding. Can you see this DNA helicase over here? It is unwinding. दिख रहा है?
Yes, sir.
Okay.
Yes, sir.
Then RNA primer for second Okazaki fragment made. Polymerase 3 binds. On top of it, you can see SSBs are also binding. And the same thing is taking place. Then further untwisting and elongation of new DNA strands. This is further opening, and you have little Okazaki fragments being made over here. The same thing. Second Okazaki fragments are done, and this is being elongated. Continuously, this goes and goes and goes along the DNA strand. Then polymerase dissociates. Process continues. Right. Then primer. This primer, you see when these little Okazaki fragments are there made, right? All of them. I draw it over here below. Five prime, 3'. 5', 3', 5', 3'. 5', 3'. So all of them have got a few nucleotide, 10, 12 nucleotides, RNA primer. These are the RNA primers which are present over here. RNA primers are present. So these primers need to be removed and replaced with DNA. These RNA primers, because we are dealing with DNA, and replaced with DNA. So these RNA primer removal gap, after removal this gap, that is filled by DNA polymerase 1. That gap is filled by all 1. Clear? Everybody, বুঝতে পারলে?
Yes, sir. Yes, sir.
Okay. পড়াচ্ছি মামা। Okay. So these but the after primer has been removed, this nick still remains, like we had seen in the former slide. This little nick this single-stranded nick still remains. Nick remains. Here, but this is sealed by DNA ligase. But for this, let's draw this once in the board. This will be much easier to draw with different color. This is the 5' to 3' direction parental strand. On top of this, you have these Okazaki fragments. These Okazaki fragments this is the 5', 3'. This yellow is the RNA primer. Now, since we have seen that DNA replication polymerase needs RNA primer to start. Later, these primers need to be removed. So what happens in the next step? These primers are removed, but this results in a gap. Primers removed, but a gap remains. This gap is filled by the action of DNA polymerase. This is the enzyme, also known as the Kronberg enzyme. This gap is filled, but still there will be a nick. Single-stranded nick joined by DNA ligase. Clear? Yes, no?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
So this is the last slide. It has got the function of different polymerases. Now polymerases have got a function known as proofreading. This ensures that opposite to A, T is there, and opposite to G, C is there. By mistake, a DATP comes in a strand where there is T. Perfect, there will be a perfect match. But instead of that, if there is a DGTP which comes, this is attached, DGTP is attached. This DNA pol 1 some people call it Kleno also. This proofreading, it will come and it will remove the false nucleotide. After removal of the false nucleotide or nucleotides, a stretch will be removed. DNA polymerase 1 will put the right nucleotide in place. So there is a high fidelity. There is a so suppose an error, when an error comes, it's catastrophic. A wrong protein will be formed. The organism may die. It will be lethal. So perfect match. So it's got a very high fidelity, and you can see that the fidelity is so high. 1 in 1 million, 1 in 10, 20,000, 1 in 30, 1 lakh, 1 in 1 crore. There may be a chance of a mistake. And therefore, the errors don't take place. It's very, very accurate. Right? Okay. So, do you people have got any questions? DNA replication? Yes, no?
No, sir.
No, sir. No questions. Okay. Okay, then Thale, I end my class over here. On Monday, we will do a class. Okay, 7 o'clock.
Okay, sir.
Okay.